Okay, hi everybody. Welcome to Chapter 17 of Bio 101 from Campbell's Essentials of Biology, 4th edition. We're going to talk about evolution of animals today. So we'll be walking through the entire animal kingdom and taking a look at how speciation occurs over time using the fossil record, DNA evidence, and biochemical evidence to sort of help us on our journey. So this is an interesting little slide. This is an '03 finding where anthropologists discovered bones on the Indonesian island of Flores dating back about 15,000 years ago of people just over 3 feet tall with heads 1/3 the size of modern humans. There was debate in the scientific community about what these bones represented. Some scientists think these bones represent pygmies of a previously unknown human species named Homo florensiens, and other scientists argued that the bones are from diseased Homo sapiens. It's difficult to know conclusively because we weren't there at the time. DNA evidence would be helpful, but it's probably extremely difficult in specimens that old to extract enough DNA to determine the actual species that these findings belong to. So let's talk about animal diversity for a bit. Animal life began in the Precambrian seas with the evolution of multicellular creatures that ate other organisms. All life here is thought to have begun from unicellular life forms, and the earliest life was thought to be the photosynthetic bacteria. The reason being that in order for life to exist, it has to have fuel, and photosynthetic organisms are able to manufacture their own fuel from the light of the sun, carbon dioxide, and water. And they produce sugars, which can serve as a fuel source to power the entire organism. The byproduct of this process is oxygen, and oxygen is necessary for aerobic respiration, cellular respiration. It's the most fruitful type of respiration in terms of energy yield. It gives us 36 ATP molecules per molecule of glucose burned, and that's enough energy to support a wide variety of life forms. So in a way, these photosynthetic organisms sort of set the stage for everything that came after. As oxygen began to accumulate in the atmosphere, the ozone layer developed, and the ozone layer, as we know, blocks the UV rays of the sun from the surface of the earth, and this allowed life to emerge from the seas and eventually evolve on land, as we'll discover in the next few slides. But in the meantime, these producers, as a result of their generation of molecules that contain chemical potential energy, specifically sugar, also set the stage for the evolution of organisms that could feed off of those fuel sources and survive. So the first heterotrophs had to come after the first phototrophs, the first autotrophs. So what we see is a change in the types of life that can evolve on this planet from simple phototrophs to simple heterotrophs and eventually to multicellular life forms, which brings us to the origins of animal diversity. So let's talk about what an animal is. Animals are eukaryotic, meaning they have a membrane-bound nucleus and membrane-bound organelles, which are special compartments in the cell that carry out specific chemical activities that allow the cell to maintain life processes. And this is different from prokaryotic organisms that don't have a membrane-bound nucleus. Their genetic material simply floats free in the cytoplasm, and they don't have as much specialization as you would see in eukaryotic life forms. So animals are also multicellular, meaning that usually they have more than one cell making up the organism. They tend to be heterotrophic organisms that obtain nutrients by ingestion. Heterotrophic meaning they derive their fuel from eating other things that contain chemical potential energy. And the ultimate source of that chemical potential energy, more often than not, is the sun, the phototrophs that end up locking down the energy from the sun into a form that other organisms can take advantage of and put to good use to perform life functions, do cellular work. They're able to digest their food within their bodies. Animal cells lack cell walls that provide the strong support in the bodies of plants and fungi and then also bacteria. So animal cells do not have this feature, but was we'll find out as a result of specialization, they have tissues that can take the role of the cell wall and do it in a way even a little bit better than the cell wall does. Most animals have muscle cells, nerve cells that control the muscles. They also tend to be diploid, meaning they have one set of genetic material from the father and one set of genetic material from the mother. As a result, most of them tend to be sexually reproducing. They also proceed through a series of typically similar developmental stages. Here you can see an example of a couple of eukaryotes. We're looking here at what looks like a python eating what appears to be perhaps a gazelle or a small deer. These are both eukaryotes, as are the plants in the background. The difference is the python and the deer are animals, and the grasses in the background are plants. They have cell walls and they're capable of photosynthesis. The green color is actually the pigment of photosynthesis called chlorophyll that is able to take UV light from the sun, capture it, and use it to generate sugar. So let's look at some of these developmental stages. This is the life cycle of a sea star as an example of embryonic development. So we'll begin here at stage 1 in meiosis. And in sexually reproducing species, the female of the species generates a gamete which contains half the genetic material of the original organism. And the female gamete is called the egg. The male gamete is called the sperm. It tends to be motile. It also contains half the genetic material of the original organism. And as a result, when these two gametes fuse, they produce a brand new diploid cell. At this point, we have all the groundwork for creating a new organism that's unique from either of the two parents because it contains a mixture of genetic material from each. And as we'll find out, or as we've already actually discussed in the chapter on genetics, the genetic material that goes into making the egg and the sperm is also shuffled during meiosis 1 when we form tetrads at metaphase meiosis 1 and we swap genetic material between homologous chromosomes. So in the sense that we shuffle the genetic deck, we actually do it two ways. We do it during meiosis in both the egg and the sperm, and we do it at fertilization when the sperm and the egg pronucleus fuse to form the zygote. Now once we have a zygote, immediately a sequence of developmental processes ensues called cleavage. So we divide from one cell to two, two to four, four to eight, and so forth. And eventually we form a mass of cells called a morula that floats free, absorbing nutrients from its surroundings. And this morula continues to divide, but it divides in a non-uniform fashion. In fact, one side of the morula will begin to divide much more rapidly than the other side, and this will form a sheet of cells that will extend down below the solid ball of cells at the superior end of this mass, form kind of a skirt. Think of a skirt around a tennis ball, okay, if you can envision that. And that skirt eventually meets at the inferior end. So imagine tying the skirt in a knot at one end, and what you end up with is a hollow ball of cells called a blastula with an inner cell mass that's eventually going to form the actual organism. But the formation of this ball is important because it sets the groundwork for a body cavity and for the formation of the germ layers, which are the cells that give rise to all the other tissues and organ systems in the mature organism. So in early gastrula, what happens is that we, instead of having a hollow ball of cells, we have an indentation on one side of this hollow ball. This is called gastrulation. And this is again a result of a unique process in development in which cells will divide more rapidly at the periphery, here and here, than in the middle, and this produces now a morphogenetic change in the organism. We still have a body cavity, but now we're beginning to move away from being a ball and more towards being a hollow disc. As the gastrula develops, eventually what happens is that we start to form an inner cell layer. This is the endoderm, an internal sac, and a future mesodermal layer, and then an ectodermal layer. These are the three germ layers of the embryo, and they give rise to all the other tissues and organs that you're going to see in the mature organism. The ectoderm tends to give rise to the epidermis and the nervous system. The mesoderm tends to give rise to the musculoskeletal system. And the endoderm tends to give rise to the viscera, the internal organs. That's sort of an oversimplification, but that's basically what these germ layers form. Now you might ask the question, well, wait a minute, sorry about that. Why is it that these germ layers only form these tissue types and they can't form any other tissue type? And that's a very interesting question, and it goes to the way that development proceeds. Cells begin development as totipotent. This zygote up here has the potential to develop into any cell type. But as the zygote matures, as cleavage ensues, and as the blastula and the gastrula are formed, what happens is that cells in different locations are getting different pieces of information as they move through time and space. And that information goes all the way to the DNA in the cells in this developing organism. Some of that genetic material remains available to the developing cells in certain regions, and some of the genetic material is basically chemically sealed off so that it can no longer be used. And that dictates the future fate of those cells from that point on. So you could think of development as going through a series of one-way doors, in which once you pass through the one-way door, it's difficult, if not impossible, to go back in the other direction because you don't have access to that information anymore and can't perform the activities necessary to generate these other tissue types. So once we get to the germ layer stage, we've essentially defined three very broad roles for the cells that remain in the organism. And from that point on, what happens is that further differentiation gives rise to all the structures that we associate with the adult animal. In large part, this is called metamorphosis, where we change form and function as we develop. And this is all driven by gene expression, where, as again, cells divide and they change where they are physically in space, and the signals that they encounter in the extracellular environment, they change their behavior, they change their shape, and they change the profile of products that they produce in order to carry out their biological function. And that gives rise to the adult organism. So here we see a sea star, fully formed and ready now to engage in another round of sexual reproduction. So, pretty interesting stuff. Now, for the longest time, most reproduction on planet Earth was asexual, where the organism would simply get to a critical mass and then divide. From one organism became two, without the need of having another organism around. And that worked pretty well for simple life forms. It allowed you to get your numbers up really high, but the problem with that mode of reproduction is that it doesn't provide any genetic diversity. And the problem with not having any genetic diversity in the population is that if that population is faced with environmental stress, it won't be able to recover. What would kill one organism would kill all the organisms. And so the evolution of sexual reproduction provided more robust species because you were able to increase the variability in the population, and as a result, increase the ability of the population of organisms to resist environmental change, to survive any type of insult. So let's take a look at some early animals and talk about the Cambrian explosion. Animals probably evolved from a colonial flagellated protist. Protist means first animal, and it's actually a separate kingdom from the animal and the plant kingdoms. It's called Kingdom Protista. And these are eukaryotes that, for most part, don't photosynthesize, don't have cell walls, and have some specialization in their organelles. So it's thought that a colonial flagellated protist, those would be a group of protists living together as a team, living in the Precambrian Sea about 600 to 700 million years ago, evolved from being a colonial life form to actually being one life form, one interdependent life form. So at the beginning of the Cambrian period, about 542 million years ago, animals underwent rapid diversification. So sort of kind of follow this if you can. We go from single-celled life forms to single-celled life forms living in groups as teams, to those teams becoming interdependent on each other, meaning that some members of the team will do certain tasks and some members of the team will do other tasks, and that group of organisms, as a result of teamwork, can survive a wider variety of environmental conditions than the organisms living independently. And then at some point that interdependence became so extreme that these organisms began not to be interdependent at all, but actually began to be part of a single organism. And so the evolution from colonial life forms to tissues was probably one of the first transitions that laid the groundwork for multicellular organisms. And as different tissues evolved, these different tissues specialized in different functions, and they came together to form organs. And as we began to form organs, those organs work together as organ systems, and organ systems work together to form an organism. And so I know that that's a lot of evolutionary time, a lot of change to take place, but remember what we talked about when we discussed Darwin, which is that natural selection is a driving force in a lot of this. When you set up conditions in which only organisms with specific characteristics can survive and reproduce, those characteristics are going to be passed on in the progeny at a greater frequency than those that don't survive. And so we can change the biological profile of an ecosystem through natural selection. So let's walk forward. Let's jump over the change from unicellular organisms to multicellular organisms, and let's talk about the Cambrian explosion. During the span of about 15 million years, all major animal body plans we see today evolved. So this was very, very rapid. Many of these animals had very bizarre shapes. You can see them here in this little figure. These are artist renditions of fossils that we've pulled out from the Cambrian period. Notice the evolution of hard parts for the first time, as opposed to plants and animals made up primarily of soft tissue. So we see some specialization here. We also see the evolution of ecosystems, where we have predation, we have production, we have mutualism, we have commensalism, and we have all of these different interactions in an environment, all of which can produce natural selection, evolutionary force. And so you see this wide variety of life forms beginning to appear. Part of what laid the groundwork for the Cambrian explosion was the production of the ozone layer, and this allowed now greater variety in the types of organisms you could produce, the body plans you could generate, because now you could produce variations in the DNA, because you weren't limited simply to organisms that could withstand the conditions of intense UV radiation at that time on planet Earth. So, a couple of theories about what ignited the Cambrian explosion. One hypothesis emphasizes increasingly complex predator-prey relationships that led to diverse adaptations to feed, move, and provide protection. And another hypothesis is that studying evolution and development, this is a branch of evolutionary science called Evo-Devo, evolution development, focuses on the evolution of genes that control the development of animal forms. So one of the things that we do with the present state of life is we like to categorize things. And we talked a little bit on Thursday about why we do this. It's not just an exercise in organization. When you categorize information, you're able to assume certain things about blocks of information because of the way you've categorized them. And I gave you the example of categorizing your mail, right? You receive mail that falls into different broad groups. You receive junk mail, you receive personal correspondence, and you receive bills. And you could approach that information two different ways. You could leave it in a disorganized fashion in a big pile, and then when you needed to extract information from that pile, you'd have to root through every single letter until you found the letter of interest. However, if you grouped those pieces of information according to characteristics, such as put all the bills in one pile, put all the personal correspondence in another, and all the junk mail in another file, which I like to call the circular file or the trash can, then it becomes much easier for you to access that information and immediately know things about it the minute you pull it out of that pile. For instance, if you go to your personal correspondence and just randomly select a letter, you immediately know that it's personal correspondence, and it probably has a greater import to you than a piece of junk mail. In addition to that, if you were called on to pull information from a particular pile, you wouldn't want to have to sort through extraneous information in order to get to the correspondence you wanted. And that's the utility of grouping things by category. It's a powerful way for you to immediately assign characteristics to an item without having to know anything else about it other than it falls into that category. And so we do that with all life on this planet through phylogeny and taxonomy, which is basically an exhaustive exercise in assigning organisms to groups based on specific characteristics. So biologists categorize animals by general features of body structure, more recently using genetic data, because the DNA revolution is really not that old. It's been probably, I would say, 35 years, but it's really taken off in the last 15 to 20. We can actually look at the DNA sequence, and information from that DNA sequence can tell us how related different organisms are to each other by checking something called homology, where basically we try and see the story the DNA tells and look for similarities. And the more similar the DNA of two organisms is, we infer the more related that they are. One branch point distinguishes sponges from all other animals because, unlike more complex animals, sponges lack true tissues. And so in the next slide here, what you see is an example of a clade. And this clade is based on a lot of different genetic data and morphological data. Morphological data is data generated based on the general body plan of an organism. And so you can group organisms according to features in the body plan. Those with similar features fall into a particular group. Those lacking those features go into a different group. This also includes genetic information of the type we just discussed, homology. The more homologous the DNA sequences are between two different organisms, the more likely they are to be more closely related, meaning that they have an ancestor, a common ancestor, that's more recent in the past than something that's more distantly related. Let's take a concrete example. Let's look, for instance, at echinoderms and chordates. Example of the chordate given here is a snake, and an echinoderm here given is a starfish. And what we find looking at this clade is that, that the chordates are more closely related to the echinoderms than the chordates would be related, say, to something like the arthropods or the roundworms, because, again, the common ancestor for the organisms that gave rise to these different phyla, these different categories, is further back in the past. It diverges more profoundly at the genetic level and at the morphological level than the common ancestor of the echinoderms and the chordates. So that's how you read these little charts that we generate. And so people who practice molecular evolution do this all day long. What they do is generate DNA sequences of the entire genome of a particular organism, and they compare it to DNA sequences of all other known organisms, and they try and generate a tree like this that shows you how related or unrelated a group of organisms are from each other. And so it gives you sort of a look back into the past as to what probably happened over evolutionary time on this planet. Okay, a second major evolutionary split is based on body symmetry. Radial symmetry refers to animals that are identical all around a central axis. An example of animals with radial symmetry would be hydras, starfish. Examples of animals with bilateral symmetry would be insects, chordates, fish, amphibians, things like that, where they have a left and a right. Here you can see an example of radial symmetry. Here they show you a flower pot, and they show you an organism with a body plan similar to a flower pot. And here you see a crustacean that has a body plan that has a left and a right half. Bilateral symmetry exists when there's only one way to split the animal into equal parts. So here with radial symmetry, we can have an infinite number of planes passing through the body plan, and each piece of the body plan would look similar, like pieces of a pie, whereas in a bilaterally symmetrical animal, there's only one mid-sagittal plane that'll define that. And so that's probably a big deal in evolution, because that's a huge leap in the body plan, in the developmental plan, that gives rise to a completely unique class of organisms that probably diverged from each other quite a long time ago. Animals also vary according to the presence and type of body cavity, which is a fluid-filled space separating the digestive tract from the outer body wall. There are differences in how the body cavity forms. If the body cavity is not completely lined with tissue derived from the mesoderm, it's called a pseudocoelom. Pseudo means fake. A true coelom body cavity is completely lined by tissue derived from the mesoderm. So again, this is a huge deviation in body plan and in body development. So we can take a look here at some different organisms that have sorry about that, no body cavity, a pseudocoelom, or a true coelom. So let's start with no body cavity. This is one of my favorite organisms. This is a flatworm. My favorite of the flatworms is the planarian. If you want to go catch a planarian, all you have to do is take a little piece of liver, put it on a fish hook, tie the fish hook to some dental floss, and drop it in your lake, pond, or local stream. Wait overnight, and you pull that liver out and put it in a little jar. And if you look in the magnifying glass, you'll see flatworms crawling all over that liver. They like liver for some reason. You can see if we open the flatworm up, you have a digestive tract that comes from the endoderm. You have a tissue-filled region that comes from the mesoderm, and a body cavity that comes from the ectoderm, but no true open space inside there. Incidentally, if you take that same flatworm and you cut it with a razor blade and you let it sit for a while along with its liver and its water, you'll find the next day that you have two flatworms. And the reason for that is that part of this body plan is totipotent stem cells that are able to regenerate the entire organism in the event of injury, which is a pretty cool trick. Wish we could do that. A pseudocoelom is basically a body cavity that is sort of incomplete. It's not lined by tissue from the mesoderm. So here you can see a muscle layer out here that comes from the mesoderm, a body covering that comes from the ectoderm, and a digestive tract that comes from the endoderm, and then the pseudocoelom is between the muscle layer and the digestive tract. Once we get to a true coelom, what you find is the GI and then tissue layers lining the coelom and suspending the internal organs from the mesoderm, and you also see a mesodermal layer and then a body covering an ectodermal layer. So this is what you see, for instance, in an earthworm. So these are big differences in body plan here, and probably indicates that the evolutionary divergence was quite a while back. Okay, let's look at some invertebrates. An invertebrate has no vertebral column. These are animals without backbones. They represent 95% of the animal kingdom. Examples are sponges. These represent multiple phyla. Sponges include sessile animals, meaning they don't move much, that lack true tissues and were once believed to be plants. The body of a sponge resembles a sac perforated with holes. The conocyte cells draw water through the walls of the sponge, and this is where it collects food. And so here you can see a picture of an actual sponge, and then you can see a zoom up of the tissue organization of a sponge. So you can pick out the flow of water, the filtering of food, and this is essentially its digestive tract, the beginning of its digestive tract. And actually, if we wanted to zoom up on that, I think we could. So why don't I give you a closer up look. So let's put this here, and then let's zoom this up a bit so you can see a little bit better. Whoops, back up, back up. Sorry about this. Just take a minute. And down. Here we go. A little bit more. There you go. Okay, and now you're starting to see what I'm talking about here. So, there's your body plan for a sponge. Let's put the actual sponge in here too so you can see. So there you can see the central cavity. These are the choanocytes in contact with an amoeba site. Flagella basically just beat, so they create the current. And these are the skeletal fibers that give it form and function. And this is the actual feeding cell, the choanocyte. And so the food is filtered out of the water as it moves through the sponge. So interesting body plan there. All right, back to where we were. Okay. Okay, cnidarians. Sorry about that. This is from the phylum Cnidaria. They are characterized by the presence of body tissues, radial symmetry, tentacles with stinging cells. The basic body plan of a cnidarian is a sac with a gastrovascular cavity, which is a central GI compartment with only one opening. The body plan has two variations, the sessile polyp and the floating medusa. Cnidarians are carnivores that use tentacles armed with cnidocytes, or stinging cells, to capture prey. And so here you can see an example of a cnidarian. This is a hydra. Okay, so here you can see the hydra itself. There's the tentacle. Okay, and what happens here is that these little tentacles, these protrusions, are made up of a coiled thread and a capsule, and they wrap around the prey, and it's the cnidocyte from which they extend. And there's usually a toxin inside these cells that will paralyze the organism so that the hydra or the jellyfish can engulf that organism into its digestive tract, its digestive cavity, and consume it. And so this is another one of these pictures where we'd like to take a bigger look. So why don't we see if we can take a bigger look here at some other body plans. Let's stretch this out. I don't know if we can stretch this out anymore. Whoops. Back up here. Let's go here. All right, let's escape out. Let's blow this up. So here you go. So here you can kind of see what we're talking about. And let's look at the hydra first. Whoops, sorry about that. Up, up, up. Up a little more. There you go. So there you can see the combination mouth-anus, the gastrovascular cavity, and the tentacles. And what happens is we paralyze our prey, we bring it into the gastrovascular cavity for eating, and then we absorb the nutrients from it. And here you can see jellyfish. Jellyfish are quite beautiful. If you ever get a chance to go to the Newport Aquarium, go to the jellyfish display. You'll be blown away by how beautiful some of these are. You can see the gastrovascular cavity, the combination mouth-anal opening, the tentacles, and essentially we paralyze the prey, bring it into the gastrovascular cavity, and dissolve it and take advantage of the nutrients. So pretty interesting system there. Okay, so let's go back and put everything where it was. I think we can shrink a little more here. There we go. Okay. All right, mollusks. From the phylum Mollusca, are represented by soft-bodied animals protected by a hard shell. Many members of this phyla use a file-like organ called a radula to scrape up food. The body of a mollusk has three main parts: a muscular foot that it uses to move, a visceral mass housing most of the internal organs, and a mantle which secretes the shell if it's present. Three major groups of mollusks are gastropods, which are protected by a single spiral shell, and bivalves, with the shell divided into two halves hinged together. And cephalopods, examples of those being octopus and squid. And so let's take a peek at some typical mollusks. And there you go. Okay. So again, you can see some typical examples. Here's your snail. There's a sea slug. There's a scallop, the octopus, and the squid. So this is where a lot of our seafood comes from, guys. This is what makes Red Lobster a good place to go eat. And if we want to zoom up on the body plan, let's do that. So let's whip around here, and let's go up there, and go across. Oh, another thing to point out with the cephalopods is they typically lack an external shell, and they're built for speed and agility. And you know this if you've ever seen a squid squirt through the water with the greatest of ease. So up, up, up we go. And here we get into the body plan. There we are. So like that. And we can shrink it a little bit. Bear with me, y'all. Okay. So what's cool about this is you start to see more specialization in the organs, right? You can see a heart, you can see a kidney, right? An anus, the gill for gas exchange. There's the mouth, there's the radula, the shell, the gastrointestinal tract, the reproductive organs, and there's the anus. And this you could think of kind of as the stomach, and this is kind of the small and large intestine all sort of lumped together. There's the coelom. There's the actual body cavity. There's the foot. And there's the beginnings of a nervous system. So you're starting to see a little bit of what makes a vertebrate here. You're starting to see organs. You're starting to see more specialization of tissues. And you can see how we're moving from a more primitive to a less primitive form. And here's a zoom up on the mouth so you can see the digestive tract, the radula, and the mouth proper. Okay, so let's hop back. Okay, flatworms. I love these guys. Phylum Platyhelminthes are the simplest bilateral animals. Flatworms include forms that are parasites or free living in marine, freshwater, or damp habitats. They have a gastrovascular cavity that's highly branched. It provides an extensive surface area for the absorption of nutrients. And here's some examples of some flatworms. So here you've got the blood fluke. Here you've got my buddy the planarian. Okay, and then the tapeworm, which is a parasite, likes to live inside the GI and feed off the food in your gastrointestinal tract. Let's talk for a minute about the tapeworm. He's got a head with a scolex and these hooks, which like to latch onto the inner wall of the GI. And then these little sections here are called proglottids, and what they contain are the reproductive organs, and they are both male and female. So every little proglottid in a tapeworm is capable of producing a brand new tapeworm sexually. If you don't remove the scolex and the proglottids hang around, you're going to get more tapeworms. So it's tough to get rid of if you ever get one. You might be wondering, how do you get a tapeworm? You eat contaminated food or water. By contaminated, we mean contains fecal material, usually from animals or from humans. So this is a problem in third world countries. Here's our buddy, the planarian. So you can see here nervous tissue has a simple brain, a little cluster of nerve cells. It's got a mouth. It's got eye spots to detect light. It's got nerve cords, and it's got a gastrovascular cavity. This is a free-living flatworm, and the blood fluke is another example of a parasite, just like the tapeworm is. He also has male and female reproductive organs, a nervous system, a gastrovascular cavity, and a mouth and an anus. So again, more specialization here. Okay, annelids from the phylum Annelida have body segmentation, where we have a subdivision of the body along its length into a series of repeated parts, has a true coelom and a complete digestive tract with two openings, a mouth and an anus. This allows one-way movement of food. The three main groups of annelids are earthworms, which eat their way through soil, and polychaetes, which are marine worms with segmental appendages for movement and gas exchange, and leeches, which are typically free-living carnivores with some blood-sucking forms. And so we start to see now even more specialization in the earthworm. One of the things that we'll notice as we look is that we have a true coelom. We also have multiple hearts. Now the heart of an earthworm is basically a beating tube. We have a mouth, we have an anus. In addition to that, we have the ability to reproduce hermaphroditically. Okay, the reproductive parts are contained in this section of the earthworm. So the earthworm can mate with itself or mate with other earthworms, depending. And it has it has a digestive tract, but doesn't really have a stomach, which means it has to feed continuously. So the life of an earthworm is basically eating dirt all day and all night in order to get enough energy to sustain an entire earthworm. But see the body segmentation here as well. Three main groups of annelids include earthworms, polychaetes, and leeches. Okay, roundworms from the phylum Nematoda are cylindrical in shape and tapered at both ends. They're the most diverse and widespread of all animals. They're also known as nematodes. They're very important in decomposition, and they can also be dangerous parasites in plants, humans, and other animals. So here you can see a free-living roundworm, and this is a parasitic roundworm, pork. This is the agent of trichinosis. And this is a canine heart infected with parasitic roundworms, heartworms. You probably heard of dogs suffering from heartworms. Is one of the reasons that we now have oral, ecto, and endoparasitics for animals. The brand my dog is on is called the Program, and it keeps it free from internal and external parasites. Okay, arthropods from the phylum Arthropoda are named for their jointed appendages. There are about 1 million arthropod species. Mostly these are insects. They're a very diverse and successful group occurring in nearly all habitats in the biosphere. There's four main groups of arthropods: arachnids, crustaceans, millipedes and centipedes, and insects. We'll go through each of these. So general characteristics. They're segmented animals with specialized segments and appendages for an efficient division of labor between body regions. The body of arthropods is completely covered in an exoskeleton and an external skeleton, and this provides protection and points of attachment for muscles that move the appendages. Now there's a consequence to this, which is that as the organism grows, it has to shed its exoskeleton in order for the soft parts to take up more room. And so for seafood, this has an important consequence. If you've ever had soft-shelled crab, this is a crab that's right after molt, and so the shell has yet to be produced and hardened. The shell is made up of a substance called chitin. And what happens in these organisms is that they're waiting now for the shell to reform after they've grown in size. So people who like to eat soft-shelled crab will fish these crab up during this point in the year and fry them whole, and you can eat them. And they're wonderful. You don't have to worry about shelling them at that point. All crustaceans do this. It's called molting. Arachnids live on land. They usually have 4 pairs of walking legs and a specialized pair of feeding appendages, and they include spiders, scorpions, ticks, and mites. And you can see examples of each of them here. There's our buddy the spider. There's a scorpion. There's another spider. There's a tick. And there's a mite. All of these are arachnids. You can tell arachnids from insects by counting the number of legs. You have 8 legs in an arachnid, and you have 6 in an insect. Okay, crustaceans are nearly all aquatic. They have multiple pairs of specialized appendages. They include crabs, lobsters, crayfish, shrimps, and barnacles. There's our buddy the crab. There's another crab. There is a crayfish. I don't know if any of you have ever seen a crayfish. You can find them around here if you go to a creek bed. They like to live under rocks in moist spaces, and they're delicious if you broil them. Shrimp, shown here. You probably are all very familiar with shrimp. Barnacles and pillbugs, believe it or not, are also crustacean. The pillbugs that we know, like roly-polies, are the kinds that you find in rotting wood, and sometimes you find them in the water in under creeks. But if you've ever gone to the Newport Aquarium, you can see very, very large examples of these pillbugs that are about the size of, I would say, a couple of loaves of bread. And they perform the same function. They're decomposers, but they live on the ocean floor. So they're quite spectacular compared to these little baby guys that we normally see around here. Okay, millipedes and centipedes. Millipedes and centipedes have similar segments over most of the body. Millipedes eat decaying plant matter and have 2 pairs of short legs per body segment, while centipedes are terrestrial carnivores with poison claws. They have 1 pair of short legs per body segment. And let me tell you what, if you've ever been bitten by a centipede, you won't forget it. It's very painful. Okay, so you want to differentiate between these two. These guys won't bother you. These are plant eaters. These guys will take a bite out of you. They have no compunction with stinging you and taking a chunk out of you. So if you see them in your house, squash them. Okay, the millipedes you can just pitch. They're not going to bother you too much. Okay, insects. They typically have a 3-part body with a head, a thorax, and an abdomen. An insect head usually has a pair of antennae, a pair of eyes, and the mouth parts, which are adapted for particular types of feeding. Flight is one key to the great success of insects. They have the ability to take to the air. And so here you can see an example from a grasshopper. There's your head, the thorax, and there's the abdomen, and there's the eye, and there are the antennae. Notice the number of legs, 6, 3 on each side. Notice the bilateral symmetry. You have a left and a right side, as you have had now for the past, 4 kingdoms? Something like that. So let's talk about some insect diversity. Insects outnumber all other life forms on the earth combined. They live in almost every terrestrial habitat, freshwater, the air, and some even like to live in saltwater. You can find them near the beach. Here you can see examples of some different types of insects. Butterflies, weevils, yellow jackets, beetles, and praying mantids. And then here's an example of a katydid. You can find members of each of these families around here if you look long enough. Yellow jackets you see a lot of times in the fall. Weevils you can find in diseased and rotting trees or plant material. We have lots of different butterflies around here. If you look in the spring and sometimes in the fall. Katydids you can find if you look. Not these exotic types, but types that live around here. And praying mantises you can find. And we have lots of beetles around here. Ladybugs are one example of a beetle. Many insects undergo metamorphosis during development. Young insects may appear to be smaller forms of the adult, or they may change from a larval form into something much different as an adult. So here you can see an example of a caterpillar basically eating and then going dormant, producing a chrysalis. Inside the chrysalis we then completely change form from a caterpillar into in this case, monarch butterfly. Monarch butterfly is famous for feeding on milkweed in its larval form, in its caterpillar form. So you can see a look at the monarch butterfly, a beautiful orange and black creature. Okay, echinoderms from the phylum Echinodermata lack body segments. They typically show radial symmetry as adults, but bilateral symmetry as larvae. They have an endoskeleton, and they have a water vascular system that facilitates movement and gas exchange. Echinoderms are a very large and diverse group. Examples are sea stars, sea urchins, sand dollars, sea cucumbers, more sea stars down here. Brittle stars are another example. Radial symmetry. Notice the radial symmetry in each of these. with the exception here, well, even the sea cucumber has radial symmetry, to be honest with you. It's basically cucumber shaped, and so you can cut it along a radial axis into many equal parts. Each of these also feature stem cells that are totipotent. So, if, for instance, if you were to take a hatchet to a sea star, cut it in half, and throw it back into the ocean, you would look back in a couple of days, you'd see two new sea stars, because in this body cavity here, we have the totipotent stem cells that can regenerate all the other sea star parts. Okay, now we get to the vertebrates. Vertebrates have a unique endoskeleton, so their hard parts are on the inside instead of the outside. That has a consequence, which is that they don't have to molt when they grow, which is pretty handy, right? They can just grow their soft parts and their hard parts at the same time. They don't have to have a period of vulnerability in which they molt and then wait for their new skeleton to grow back. So their endoskeletons are composed of a cranium, a backbone made of a series of bones called vertebrae. And you're looking here at a typical vertebrate. This is a rattlesnake. And you can see the cranium, and you can see the vertebral column, and these are all ribs here. Lots of ribs. And inside the vertebral column is a spinal cord, and inside the cranium is your brain. So let's look at some chordates. Chordates all share four key features that appear in the embryo and sometimes the adult. They have a dorsal hollow nerve cord, right there. They have a notochord, this structure here. They have pharyngeal slits. They have a post-anal tail. Okay, so there is a tail, and there's the anus. Okay, so the tail comes after the anus. Another chordate characteristic is body segmentation, which is apparent in the backbones of vertebrates, the segmental muscles of all chordates. Chordates consist of three groups of invertebrates. Lancelets are blade-like animals without a cranium. Tunicates or sea squirts also lack a cranium, and hagfishes are eel-like forms that have a cranium. All other chordates are vertebrates. And so here's an example of some chordates. These are tunicates, and this is a lancelet. Okay, it has a mouth, has a tail, has a notochord. Looks almost, and I'm kind of getting ahead of myself here, like a fish. Okay, so if we move up a little bit evolutionarily, we're going to start to see almost a full body plan. So here is an overview of chordate and vertebrate evolution. So the ancestral chordate had a common ancestor that gave rise to the early tunicates. So this is the most primitive member of this branch of animals. A little bit more recently, a common ancestor diverged that gave rise to the lancelets, and then to the group that gave rise to the hagfishes, and this other subgroup down here, which had another common ancestor, diverging even more recently ago, that gave rise to the lampreys, and then another common ancestor even more recently that gave rise to the cartilaginous fishes like sharks, and then another common ancestor that gave rise to the bony fishes, and another common ancestor even more recently that gave rise to the amphibians, and then finally another common ancestor that gave rise to the reptiles and the mammals. Okay, so in terms of how ancient evolutionarily each of these life forms are, we would go the most primitive organism here and the more primitive organisms as we descend down this clade. And the way that we know this is that we compare DNA and we compare body plans. So, and here you can see the different groups, right? The chordates envelop all of these. Okay, these are all chordates. The vertebrates envelop this group. Okay, they have a vertebral column. And the tetrapods, four-legged creatures, this group. And the amnivoles, the reptiles, and the amphibians. So let's look at the fishes. The first vertebrates were aquatic and probably evolved during the early Cambrian about 542 million years ago. They lacked jaws and are represented today by the hagfishes. And so here you can see an example of a hagfish, a jawless fish. Produces slime on its outer body covering, probably a way to escape predation. Another member of this group are the lampreys. They're vertebrates, they have a cranium, but they have no jaws. You've probably seen lampreys hitching rides on the backs of other marine creatures. Okay, they essentially live as parasites. They feed off food that is near the creature they're hitching a ride on, and they don't really have an ability to live totally independently of another organism. They tend to have a parasitic relationship with their host. The two major groups of living fishes are the cartilaginous fishes, which are the sharks and the rays, which have a flexible skeleton made of cartilage, and the bony fishes, with a skeleton reinforced by hard calcium salts. The bony fishes include ray-finned fishes and lungfishes and lobe-finned fishes. Cartilaginous and bony fishes have a lateral line system that detects minor vibrations in the water. This is part of the nervous system. To provide lift off the bottom, the cartilaginous fishes must swim, but the bony fishes have swim bladders, which are gas-filled sacs that make them buoyant. So here you can see the different groups, right? There's our hagfish, there's our lamprey, there's our cartilaginous fish, and here's our bony fish. And you can see here the lateral line system in each of these. So this is our most advanced member, next most advanced, and least advanced. Okay. Now, the first life forms to get out of the water were the amphibians. They're able to live both in water and in land, and they can breathe either with lungs or they can use their skin to exchange oxygen between the blood and the external surroundings if they're in water. Amphibians exhibit a mixture of aquatic and terrestrial adaptations. They usually need water in order to reproduce, although not always. They typically undergo metamorphosis from an aquatic larva to a terrestrial adult. And here you can see some examples of amphibians. Here's a tadpole and an adult golden palm tree frog. This is the Texas barred tiger salamander and a red-eyed frog. Amphibians were the first vertebrates to colonize land. They descended from fishes and had lungs and fins with muscles in them. So lobe-finned fishes are thought to be the direct ancestors of the early amphibians. Terrestrial vertebrates are collectively known as tetrapods, which means 4 feet. 1, 2, 3, 4 on the other side. Tetrapods include amphibians, reptiles, and mammals. Reptiles, including birds, and mammals are amniotes, which produce amniotic eggs that consist of a fluid-filled shell inside which the embryo develops. Reptiles include snakes, lizards, turtles, crocs, alligators, and birds. Reptile adaptations to living on land include amniotic eggs and scaled waterproof skin. Here's an example of the reptiles. Let's take the present-day ones first. Snakes, lizards, turtles, crocs, and birds, which I know it seems unusual to include them in this kingdom, but we now have fossil evidence that shows that birds are probably the direct descendants of dinosaurs, and that dinosaurs are probably the ancestors of some, if not all, of these present-day groups. Non-bird reptiles are ectotherms. They're sometimes referred to as cold-blooded, which means that they obtain their body heat from the environment. So they assume the body temperature of their surroundings. A non-bird reptile can survive on less than 10% of the calories required by a bird or a mammal of the equivalent size. Reptiles diversified extensively during the Mesozoic. Dinosaurs were the largest animals ever to live on land. Recent genetic evidence shows that birds evolved from a lineage of small two-legged dinosaurs during the great reptilian diversification, called radiation, of the Mesozoic era. Birds have many adaptations that make them lighter in flight. They have honeycombed bones, so they're not very dense, 1 instead of 2 ovaries, a beak instead of teeth. Unlike other reptiles, Birds are endotherms, maintaining a warmer, steady body temperature. Birds' wings, adapted for flight, are airfoils powered by the breast muscles and anchored to a keel-like breastbone. So here you can see an example of how we achieve flight. Now you might wonder, what in the world is this diagram showing me underneath this flying eagle? Well, it shows you how it is that many organisms that fly through the air are able to get off the ground. The shape of the wing is critical. Air tends to flow in streams. This is called the laminar flow theory. And the idea behind an airfoil is that if a moving object is cutting through the air, the density of the air on top of the airfoil is going to be less than the density of the air underneath it. And as a result, it's going to provide lift as the animal accelerates forward, because there's less pressure here and more pressure there, which results in a net force in the upward direction. This is why the wings of airplanes are curved in this same fashion. Mammals are the first true mammals arose about 200 million years ago, and they were probably small nocturnal insectivores. Most mammals are terrestrial, although dolphins, porpoises, and whales are aquatic. Mammalian hallmarks are hair and mammary glands that produce milk, which nourish the young. There are three major groups of mammals. Monotremes, which are egg-laying mammals, so they have external development. Marsupials, which are pouched mammals with a placenta. And eutherians, which are placental mammals. Eutherian placentals provide more intimate and long-lasting association between the mother and her developing young than do marsupial placentas. And so here you can see examples of the egg layers, the monotremes, the marsupials, such as the kangaroo, and placental mammals, such as tigers, bison, lions, bears, and so forth. We are also placental mammals. Mammals, again, because they have mammary glands that produce milk to nourish the young. Humans are primates. This mammalian group also includes lorises, podos, lemurs, tarsiers, monkeys, and apes. And so you can see examples of each of these here, okay, and the subgroups in which they fall. Primates evolved from insect-eating mammals during the late Cretaceous. They're distinguished by characteristics that were shaped by the demands of living in trees. This is called arboreal lifestyle. These characteristics include limber shoulder joints, eyes in the front of the face, so binocular vision for depth perception, excellent hand-eye coordination, and extensive parental care. Taxonomists split the primates into three groups. The first group includes the lorises, the podos, and the lemurs. Tarsiers form the second group, and the third group, the anthropods, includes monkeys, hominids, and humans. And so here you can see examples of each of these. If you go to the zoo, you can find all of these, and it's very interesting to watch them behave and interact. There's lemurs, tarsiers, podus monkeys. These are called Old World monkeys because you find these primarily in Europe. New World monkeys you find primarily in South America. Gibbons, orangutans, chimpanzees, gorillas, and humans. We're most closely related genetically to the chimpanzee, but as we'll see in a moment, that doesn't mean the chimpanzee was our ancestor. Humans and chimps have a shared common African ancestry for all but the last 6 million years or so. Chimpanzees and humans represent two divergent branches of the anthropod tree that each evolved from a common, less specialized ancestor. Our ancestors were not chimpanzees or any other modern ape. Human evolution is not a ladder with a parade of fossil hominids leading directly to modern humans. Instead, human evolution is more like a multi-branch bush than a ladder. At times in hominid history, several different human species coexisted. Upright posture and an enlarged brain appeared at separate times during human evolution. Different human features evolved at different rates. And here you can see some examples of some of the early human species. And so a lot of these came into and out of existence and didn't contribute directly to the lineage that led to human beings. The earliest to stand erect was Australopithecus africanus. And this gave rise to some subsequent hominids, Paranthropus robustus, Homo habilis, and finally Homo erectus, and then Homo neanderthalis, and then finally Homo sapiens. Homo erectus, we don't know how many millions of years ago they went extinct. Homo neanderthalis and Homo sapiens, as you can see, coexisted for quite a long period of time. And eventually what happened was that the Homo sapiens species ended up being the predominant species, but it's not out of the realm of probability that they interbred. Okay, and so this is probably what produces some of the racial differences that we see today. Longer ago, we can see Australopithecus going extinct. This is considered to be one radiation of the human tree that sort of went off into no man's land, as opposed to the line that gave rise eventually to Homo habilis and erectus. So how each of these radiated from each other, it's hard to say. If we look at Australopithecus, before there was the genus Homo, several hominid species of the genus Australopithecus walked the African savanna. Fossil evidence pushes bipedalism of Australopithecus afarensis back to at least 4 million years ago. And so here you can see a modern artist's conception of an Australopithecus afarensis male. And here's some ancient footprints showing bipedalism. And here is some skeletal remains, actual skeletal tissue. Homo habilis had a larger brain, was intermediate in size between Australopithecus and modern humans, walked upright, and made stone tools that enhanced hunting, gathering, and scavenging on the African savanna. Homo erectus was the first species to extend humanity's range from Africa to other continents. The global dispersal began about approximately 2 million years ago. Homo erectus was taller than Homo habilis, had a larger brain, and gave rise to Neanderthals. So one of the questions we've asked is that since Neanderthals are one of our most recent ancestors, we shared a common ancestor, geneticists have noted that the gene, well, the question is, what did Neanderthals look like? And geneticists have noticed that the gene MCLR has a large effect on human hair and skin pigment. So the question is, what form of the gene will be found in Neanderthals? And so the pigment gene was isolated from Neanderthal DNA from fossil remains. And the results were that the Neanderthal gene contained a mutation not found in modern humans. Human cells engineered to carry the Neanderthal version of the gene reacted in ways that suggest that the Neanderthals had red hair and pale skin. So very different from the way we generally conceive the appearance of Neanderthals from popular media. So, the oldest known fossils of our own species, Homo sapiens, were discovered in Ethiopia, dating from about, let's call it 180,000 years ago or so. DNA studies strongly suggest that all living humans can trace their ancestry back to a single African Homo sapiens woman who lived about 180,000 years ago, again from DNA evidence, and particularly mitochondrial DNA evidence, because you get all of your mitochondria from your mother. So we're able to trace the maternal lineage simply by looking at mitochondrial DNA. Fossil evidence suggests that our species emerged from Africa in one or more waves. The oldest fossils of Homo sapiens outside Africa were 50,000 years old, and the oldest fossils of humans in the New World are uncertain, but are at least 15,000 years old. So how has our culture evolved in this period of time? Culture is the social transmission of accumulated knowledge, customs, beliefs, and art over generation. Culture is primarily transmitted by language. Cultural evolution has 3 major stages. First, nomads, who are hunter-gatherers. They made tools and they organized communal activities, and they divided labor, and they also created art. Part of this is the evolution of the opposable thumb, which allowed them to create tools and to have language and written communication and artistic communication. The second main stage of cultural evolution was the development of agriculture in Africa, Eurasia, and the Americas about 13,000 years ago. The third stage was the Industrial Revolution, which began in the 1700s. The global consequences of human evolution have been enormous. Humans can defy our physical limits, shortcut biological evolution, and change the environment to meet their own needs. So let's look at recent human evolution. Biologists have identified a number of genes that appear to be undergoing rapid evolution. One such is the FOXP2 gene, known to function in speech. Studies of this gene indicate that the human form likely arose within the last 100,000 years. Neanderthals had the same version of FOXP2 as modern humans. And so this is one of the indications that they are more closely related to us than any of the other fossil ancestors. Okay, with that, we come to the end of this chapter, and I will see everybody Thursday night, and you can punch us up on YouTube and watch it at your leisure. I appreciate your attention, and I'll see you in class.